Our next speaker is Erin Mckenna, who is a graduate student at University of Washington in Shendure Lab. And he's a former Brody, having done comp bio work in Gadi Getz's lab. And he will share his recent work on lineage tracing with CRISPR-Cas9.
Yeah, it's so great to be back. It takes leaving the Broad to get to the big room. So for all of you Brodies, if you come back, you can speak in the big room. So today I want to tell you about our work on whole lineage tracing using the CRISPR-Cas9 system. And this is done in the Shendure Lab at the University of Washington in the Department of Genome Sciences. And our overarching goal is to do whole organism lineage tracing. And what does that mean? Well, lineage tracing is determining the relationship between a cell and its progeny. So you can imagine here if we say selected this glowing red cell and it led to the large portions of the heart, we'd be able to connect them with lineage tracing. And of course, we can't really mention lineage tracing without mentioning the kind of tour de force of especially whole organism lineage tracing, and that is the full lineage of C. elegans. And this work was monumental, done in the late 70s and early 80s by Sulston and colleagues. And what they were able to do with this model organism is trace the trajectory from the single cell zygote all the way to the fully formed worm using simple light microscopy. And so up here you can see there we go. You can see the C. elegans, which has 800 to 1,000 cells, depending on the sex. And you can see here on the left some of Sulston's early drawings relating individual cells and their progeny. And this effort went on for many years, carefully watching C. elegans under the microscope. And what they were able to do at the end is generate this full fate map, or full whole organism lineage tree from those collected data. And this is an absolutely beautiful and monumental task that they accomplished, but they had certain advantages that we may not have. So C. elegans, as I said, has 800 to 1,000 cells. It's transparent throughout its life. So you can simply watch it under a microscope. And most importantly, the lineage is invariant. So if you take any single C. elegans off the plate, look at it under a microscope, those exact same cells will divide in the exact same ways. So you can kind of repeatedly sample and build up these really wonderful lineage trees. So in invertebrates and in mammals, we're not so fortunate. Most vertebrates become opaque early in development. The developmental trajectories at least have some stochastic nature to them. And most importantly, right, the numbers are exponentially bigger. So the estimate in an adult human is that we have about 37 trillion cells that emerge from that single cell zygote. And so you can't use the same techniques. So the field over the last 100 years has come up with a whole bunch of different strategies to tackle this, but they all come down to the approach of marking individual cells with some kind of marker, be it dyes at the turn of the 19th century, fluorescent markers in the kind of modern era, to even the more advanced techniques like rainbow. But the challenge with all of these techniques is that they're static, right? So you've marked a cell. So you've marked this orange cell down here, and all you can do is say which cells then become orange in the final animal. There's no relationship between cells, so you can't say anything about maybe these two cells versus these two cells. And so we set out to kind of change this, and of course you can keep doing this again and again for different cell types and build up these more comprehensive lineages, but it's obviously very tedious and it takes a lot of work. So we set out to devise a system, and as you can imagine, it's going to be based on CRISPR. But we set out with certain design goals in mind. We wanted to be able to distinguish between the individual cells in a vertebrate or a mammal, so it needs to have a large information capacity. We need to be able to distinguish between cells and their progeny. So these barcodes we developed had to pick up editing over time. We wanted, of course, we're a sequencing lab, so we wanted to be able to measure this using second-generation sequencing. And importantly, we don't want to mess with normal development. And we would like to, as these edits accumulate over time, be able to disentangle cells that may look genetically similar, like say from somatic mutations, but we have marks that can distinguish them. So as you can imagine, we took the CRISPR-Cas9 approach, and I'll tell you kind of 3 quick vignettes about what we did. First, how we did it using CRISPR and CRISPR arrays. And then secondly, how we reconstructed Lineage in a kind of a fake cell culture system, or simulated cell culture system. And lastly, how we applied this to whole organism lineage tracing in zebrafish. So all the parts here are going to be very familiar to everyone in the audience. We have Cas9, we have our guide, and then in the genome of interest, we introduce a series of target sequences for this guide. Cas9 comes along, NHEJ, or Alt-NHEJ, resolves this double-stranded break into an insertion or a deletion. And so in that way, this target sequence starts acquiring information. So our first approach was, well, we needed a whole bunch of these targets. Where do we get targets that we know work, and especially with the constraint of using a single guide initially? So we turned to the GuideSeq paper, and we took one on target here in bright red, and then tiled a series of progressively weaker off-target sequences. So the guide will have the most preference for this first target and then kind of fall off as you go along the target array. We put these into 293 T cells. We stably integrated it with lentivirus, and then we hit them with Cas9 in the guide. And well, we're of course curious to see if this actually was going to work. And so I'll show you a bunch of these plots today, and I just want to orient you before we get too far into it. So you can imagine that series of 10 targets. Each of those here is represented by this light gray box. Next to it is the PAM sequence in dark gray. And what you're seeing on the top here is a histogram of editing over all of the sequencing reads we recovered. And you can see here in this particular instance, we see almost 50% editing at this first site, and it kind of bounces around a little bit out to the 5th or 6th site. And also in blue is the insertions. Unfortunately, we didn't have any insertions in this example, so taking my word on it here. And you can see here on the bottom, what we're showing is this first read essentially is all wild type, so we've colored it green. And then in the second read, we have a 6 base pair deletion at that first site. And so we collected those cells, those 293 T cells, and we want to see, do we actually get some diversity in the editing? So here's one example experiment. We see a large amount of editing, and it's somewhat diverse. We see mostly, of course, editing at this on-target site, but then we do see editing going out to the more distal off-target sites. And you can ask a simple question like, how many of these, if you're interested in the information content, how many of these sites are affected by any of these edits? So these light pink bars here in the histogram are indels that only affect a single site, whereas these red ones affect, or light red ones affect 2, 3, and so on. So we're at least encoding information mostly at a single site, although we do get kind of multi-site dropouts, which unfortunately could, say, delete an informative deletion here, but in general, those ends of those double-stranded, or those large deletions are also informative. Okay, so we have editing in cells, and it seems to be generating some diversity. What can we do with it? So the first thing we turned to was trying to reconstruct lineage in cultured human cells. And to do this, we made a kind of synthetic lineage. So what we did is we took an unedited ancestor cell, one of those 293 T cells. We performed one round of editing. We sorted it out into 12 subpopulations, of which 7 of those had edits. So these are the 7 populations that kind of survived our filter. We then re-edited those subpopulations independently and sorted each of those into populations of 100 cells each, so 2 replicates of 100 cells each. And what we wanted to be able to see, right, is can we relate these daughter cells to their parental line? And can we disentangle one daughter cell parental line from another, say blue from green here? And I just want to show the blue lineage because it's a nice example. So in this in the ancestral population, we see this 6 base pair edit at the first site. And then in the subsequent editing in the daughter populations, we see both editing increase at kind of the more off-target sites, but also a diversity of editing is growing. And importantly, we can put this all back into a big tree using maximum parsimony, and we can ask how many populations were we able to correctly assign. And so we make the claim that 99.7% of the distinguishable single-cell colonies we could assign back to their correct clade. That's, of course, caveated here. This orange cell population, we actually ended up sorting 2 cells, one of which was wild type and one of which had a founder edit. And you can actually see that here. These orange guys have the founder edit from one of the cells, and then these guys with kind of mixed all over seem to have that wild type and then subsequent editing. So it's a good point to pause and say a method isn't a method until you have a name. So at this point, we were pretty confident it might work. And all credit goes to Greg, one of my fellow co-first authors, to coming up with the name Gestalt, which is genome editing of synthetic target arrays for lineage tracing. So we knew we could go forward. We had a working system. We had a name. Could we test this out in actual living systems? So it's a little washed out here, but so you can imagine taking this into a vertebrate system, and we chose, because of collaborators, the zebrafish. And so our collaborators grew up a Gestalt positive zebrafish, crossed that with wild type fish. And then what we did, and what was different than the cell culture work, is we really wanted to up the diversity that we saw in these target arrays. So we switched strategies. And instead of using this one guide with many off-targets, we switched to RNPs with a single guide for each target site. So for instance, this blue matches this blue completely, yellow matches yellow, and so on. So we did this for a 10-target array. And the hope was that we would get a larger and more diverse editing pattern. And so we took these RNPs, and the nice thing about working in zebrafish is you can just inject this right into the single-cell zygote. And then you can sample those embryos over time. And we took a whole bunch of different time points, really early in development through kind of the canonical developmental stages, and then harvested 2 adult zebrafish. And I won't talk about, for the sake of time, I won't talk about the embryo work, so if you have questions, let me know. So from the adult here, we sampled 11 organs, or 11 organs and tissues. And the nice thing about these fish was that they had a cardiomyocyte marker, so all the cardiomyocytes would glow green. So we could take the heart, disassociate it into populations, flow sort it, and pull out the cardiomyocytes. So we did this for our first adult zebrafish, which I'll talk about today. And we sampled almost 200,000 cells. We know they're cells because we used UMIs, so unique molecular identifiers, to tag each cell's product. So we recovered almost 200,000 cells from the whole fish. And the diversity here is about 17,000 different alleles, so unique editing outcomes over the full target array. And if you go in and look at individual organs, so for instance, here is the blood, we see a very different story than what we saw in the cell culture system, right? Where we had one target and then a series of off targets. These are all on targets. And you can see over the full array, we get a really diverse set of edits. And there's some other really interesting things that started to pop up. We didn't even know if this was going to work to begin with. But we started to see some really interesting biology. First and foremost, you can see here these starred alleles. This is numbers of cells on the log scale. So the first 5 alleles in the zebrafish blood seem to make up about 98 to 99% of all the alleles we recovered from the blood. And you can imagine that with this RNP system, with editing coming on relatively early, you'd expect a much larger diversity of editing patterns to persist in the blood. So there must have been some kind of collapse of the alleles we've seen, whether it's clonal dominance or it's hard to speculate, but it's certainly interesting. And it was an important lesson. This is actually the second iteration of FISH we did because we didn't sequence blood in the initial iteration. And what you'll see is that blood is everywhere. And this, of course, should be obvious to everyone, but we didn't think about it before we dove in. And so you really do need to sequence blood if you want to understand the whole fish. And you can see this if you look, say, in brain, one of our more diverse organs. You can see there's alleles here that are specific just to brain, but there's a series of alleles that exactly match those blood alleles. And importantly, they're actually in rank order with the blood alleles. So this, obvious blood is flowing through all these organs. Our dissections aren't perfect. What we managed with picking up was pre-sequencing the blood. We know what to expect for blood alleles. And we see this in all of the organs that we sampled. We looked, here are the 5 predominant blood alleles, and you can look across all of the different populations and see those blood alleles in different proportions, but also in the same consistent rank order. And importantly, a great milestone for us was that we didn't see any of these blood alleles in the cardiomyocytes, which, given that they're flow sorted on GFP, should be a relatively pure population. So it really gave us a lot of hope that this was working as expected. So, of course, we sampled all these organs. What can we do with it? And of course, the cell lineage, or the synthetic lineage, kind of hints at what we're going to do. We want to combine all of these organs into a single whole organism lineage tree. So borrowing from phylogenetics, we used maximum parsimony to combine all of these alleles together using a Kamem-Sokal model, if anyone's interested. And we filtered the tree you're about to see for alleles that were seen more than 4 times. And you'll see why, just because it's rather large. It doesn't come out great on the PDF, but you can see this enormous tree that we recover from all of the alleles in the zebrafish. And it's a little too much to take in here, but you can see that there's the organs here are colored by their proportion, and here you can see all of the alleles that we recovered at least 4 times. But let's zoom in a little bit and make this a little easier to see. So let's take this very first clade, and what we wanted to know was, does the clade reduce in complexity as we start adding additional edits, right? So does it go from having all germ layers down to having single germ layers or even having single organs? So we'll take this first clade here that's defined by this kind of large deletion, I think between sites 5 and 6. And we can see a wide diversity of both germ layers and organ types that we recovered. But you can start adding additional editing onto this. So here we've added A, B, and C edit. And you can see that the kind of different organs and germ layers are slowly reducing. And just to make it a little easier here, we'll focus on 1B. And you can see now we're down to essentially just eyes and parts of the heart. And so you can even go further and reduce down more. And you can see in this kind of tiny subset about 2,000 cells that make up completely just the eyes, so left and a right eye in about equal proportion, which gave us faith in this proof of concept that we're actually capturing real biology. So I've told you about our approach and how we edited, how we used CRISPR-Cas9 to reconstruct lineage, and how we used it in zebrafish. And of course, this is just the beginning, we hope. There's a lot more to do. We can integrate this with transcriptomic data, single-cell technologies. There's applications, of course, in tumor heterogeneity or tumor growth in general. And we're excited, of course, to start rolling this out to additional model organisms. So as I said before, this is a joint project. This is one of the most fun projects of my life. Greg Finley in our lab and James Gagnon here, along with the Marshall Horowitz lab, the Shendure Lab, and of course Jay Shendure, my mentor. And this it's a great time to give a shout out. This of course wouldn't be possible without the contributions of Addgene, all the labs who have contributed to Addgene, and of course our funders. So with that, I'll take any questions. Sure. I mean, there's many interesting follow-ons you could do. I mean, you can do essentially pulsed. This is all, in the zebrafish, it was all done with RNPs, right? So you can imagine integrating that, whatever Cas9 and its promoter into the genome. You can imagine doing TET-on, a whole bunch of other things. And I think we're actively working on kind of technological improvements as well. But great question.
It's a nice talk. So I had a question about, obviously the system relies on making double-strand breaks with
Yes.
DCAS9. And the DNA damage response in some organisms is really regulated differently during development. So do you worry at all that by making these breaks, you're making subtle changes to the developmental structure itself?
Yes, of course. I mean, cells endure a lot of double-stranded breaks in development in general. And looking at, I mean, there's the kind of metric in zebrafish is to, take one embryo that's mutated in some way and hold it up against an unmutated one. And these guys looked healthy and hit normal developmental milestones, but totally. And there could totally be organ systems that are more affected. So some of that's things we have to kind of work out and really play with a little bit more. But great question.